Okay, I'm going to continue talking about some of the various immunoassays, but now I'm talking more about um, how they can be used and what you can do with them. So, first on the list is precipitation reactions in fluids. So, if you have a culture or a colony of a particular kind of bacteria, let's say Klebsiella, and you have anti Klebsiella antibodies, you can mix the two together, and if it clumps up, you can start seeing whether or not you're getting an antibody antigen reaction. So it's sensitive to down to about 20 micrograms of antibody per milliliter. So it takes quite a bit of antibody for it to actually work. Precipitation reactions in gels, you can do what's called the Ochterlony test, and it's a double immunodeficiency so that you get that smiley face, clear zone, cloudy zone in agar media. There's other direct agglutinations, um, agglutination reactions, and they can be much more sensitive, especially in small volumes. Probably the most sensitive is the radioimmunoassays, and you use that to test for specific antigens in the bloodstream. So I could pull a blood sample and test for, or a urine sample and test for um, prostate-specific antigen in urine, and be able to tell you the exact, pretty close to the exact levels of prostate-specific antigen that is in the urine sample. So they can use it to detect very small amounts of uh, proteins. Enzyme-linked immunosorbent assays are really sensitive and they're great tools. They've been using them for years. You can measure either the amount of antigen or the amount of antibody in a sample. And um, the the good thing about the ELISAs, um, it's enzyme linked, so the enzyme linkage gives you a colorimetric change. So you can measure colorimetric changes and be able to tell whether or not you're getting a cross reaction occurring. Um, you can run a 96 well plate and probably get 80 to 85 samples on a plate and run each one in duplicate. So technically, you could probably get 40 different samples on a single plate. So if they're testing blood samples for lupus, or they're checking blood samples for Lyme's disease, they can send the reference sample off to a lab that runs this type of assay, and they can run a bunch of them at one time. So a plate that costs $500 to run, you're getting 40 tests out of it. So it really is pretty efficient. Now they've added chemioluminescence and fluorescence to the ELISAs in order to be able to detect levels, and it gives it a little more sensitivity. Immunofluorescence can be used directly to stain the bacteria or to stain a specific kind of cell, like if you're looking for cancer cells, like if you're looking for cancer cells in a tissue sample, you can stain it and see where the cancer cells are located. Flow cytometry, you can actually separate out cancerous cells or cells carrying a specific CD protein on their cell surface. So a lot of times you have to get the right combination of agglutination. So in this case, they're using sheep red blood cells, and they're looking for it to aggregate together when they add the anti-sheep red blood cell serum, and then you get a pattern that shows positive hemagglutination starting at tube 3. You can actually radio label using radioactive iodine, which is kind of dangerous to work with, so not many labs like to work with it. Radioactive iodine can concentrate in the thymus gland and cause problems, so you want to be really careful. But you can take cytokines and bind them, label them with radioactive iodine, bind them to the bottom of a well, and tell the exact concentration of unlabeled cytokine that's in a sample.